So let us start with today's episode of plasmolysis and deplasmolysis. So what are the learning outcomes of today's episode? Today's episode is very small, so we will be dealing with only plasmolysis and deplasmolysis. Obviously with diagram, we should know what is happening to the cell, so we will go through its diagram also. So it is with diagram we will be discussing plasmolysis and deplasmolysis. So let us start with plasmolysis. Now here comes the main topic of today, the plasmolysis. Now what is it? Now plasmolysis happens when you are placing the cells in a solution which has a higher concentration of solute than the cell itself has. So what is that type of solution called? Hypertonic solution. So students, I feel you have gone through the lesson of tonicity which I have already taught earlier. This causes the protoplasm and all the material on inside of the cell to shrink away from the cell wall. We will see in diagram also what is happening. So what is happening here? The cell starts shrinking in plasmolysis. So when it is placed in hypertonic solution, all the water from inside the cell, it move outside. They lose water and it is shrinking away from the cell wall. So let us see the diagrammatic representation of this one. So let us see here what is happening. This is a turgid cell or a normal cell. We can say this is the vacuole. Now this cell is in normal condition. Now as soon as we are keeping it in a hypertonic solution, that is a solution having solutes more than that which is present inside the cell. So water starts oozing out. And gradually we can see the cell wall and the plasma membrane, they are having a space in between two. So it is successive stages of shrinkage of the protoplasm. So protoplasm is shrinking from the cell wall after being placed in a hypertonic solution. Now this is what is happening in a plasmolyzed cell. The cell wall is rigid, but the protoplasm and all, they have shrunk. So that there is a shrinkage of protoplasm and this is a plasmolyzed cell. Now these are the stages of plasmolysis. So B, C and D, these three are the stages of plasmolysis. Now let us see what is the opposite of plasmolysis. Obviously it will be deplasmolysis. So in the next slide we will be studying that. Now here comes the next topic, that is deplasmolysis. What is happening here? It is the reverse of plasmolysis, and there was a shrinkage of cell when we were placing it in hypertonic solution. Remember that, dear students? Now here it will be swelling of the plasmolyzed cell under the influence of hypotonic solution or water is called deplasmolysis. So it is exactly the opposite what was happening in plasmolysis. Now it is only possible in a living cell and its cytoplasm is not dead or damaged. If it is dead or damaged, we can't see the changes in this cell. Now what happens in this deplasmolysis? Water from outside the environment rushes into the cell through the process of osmosis, and the cytoplasm as well as the plasma membrane, they push against the cell wall, thereby helping the plant to stand upright again. So this deplasmolysis helps all the cells to regain their normal condition and it helps the cell or the plant to stand upright. So previously we have seen that plasmolysis causes shrinkage of the cell in hypertonic solution. This is causing swelling of the cell, plasmolyzed cell in hypotonic solution. And all the living cells, when they are placed in hypotonic, they will be simply in cytoplasm. If it is not dead, they will show the changes like this. Plasmolyzed cells actually, not the normal cells. The cells which are already shrunken or the shrinkage has already occurred in those cells only. If we are placing them in hypotonic solution, they will show these changes. and the water from outside will move in, and the cytoplasm as well as the plasma membrane push the cell against the cell wall, and they'll again come back to the same upright position. So let us see in diagram what is happening here. Now here see the difference. Plasmolysis, all the water was going out when placed in hypertonic solution, and when this plasmolyzed cell is again placed in a hypotonic solution, the water from outside gushes inside, because now the concentration of solutes inside will be more than that outside. So is it clear to everyone? The concentration of the cell inside will be more than outside. So water from outside is gushing inside. So students, can you just tell me what is happening here? Recall and tell me what is the process. We have read like this only in one of the previous videos. I think in osmosis you have read that. So this is what is similar to exosmosis, and this is what is similar to endosmosis. So I think it is clear. So this is the exosmosis happening, that is water is oozing out of the cell, and this is what endosmosis is happening, that water is moving inside the cell. Now we will see the difference in a tabular form. It is very important for your ICSE exams, because all these differences between turgidity, flaccidity, diffusion, osmosis, frequently now and then you will be asked in your question, in your tests, either school tests or your board exams. They'll be asking you the differences between plasmolysis, deplasmolysis, or I have discussed in your previous one like diffusion, osmosis, diffusion and imbibition. So till now, if you have not gone through all my videos, please do go through it. Subscribe to my channel. Don't forget to like and share, and so that you can keep on getting my updates. I have told you that generally I'll be posting twice a week. So please stay updated with all my videos. Now let us see what is the difference. Now let us come back to topic again and see the difference between plasmolysis and deplasmolysis. Now here comes the basic difference between plasmolysis and deplasmolysis. Now it is the shrinkage of the cytoplasm from the cell wall, and it is the swelling of the plasmolyzed cell such that it presses against the cell wall. So I think it is very much clear, as I am teaching it in a very simple way. Now next comes the type of solution you should keep in. Now cells when kept in hypertonic solution will give you a plasmolyzed cell, and when the plasmolyzed cell is kept in a hypotonic solution such as pure water, you will get a cell which is regaining its normal form and shape. So you will regain a nice cell which is back to its normal form, that is called deplasmolysis, and it is the result of exosmosis, and it is the result of endosmosis. So is it clear to everyone? Dear students, if you are having any doubts, any queries regarding today's topic, please do ask in my comment box, and I'll be answering all your queries in the next video. So this is all for today. Go through your textbook, whatever book you are following, or if you are not clear, go through my videos again and again. I think you will be quite clear in that. So we'll be meeting very soon with the next part of plant physiology. That is, we'll be studying about absorption of water and minerals by the roots. Thank you.